Hi all, good evening. Welcome back to the live. Firstly, I need to say sorry lah because actually the live is start on 5 p.m. because of the personal issue. So I delay the live for 1 hour. So now it is 6 o'clock. So I will continue the topic gene expression part 2 about the chapter 17 inheritance and genetic control. Okay, so chapter, I will proceed to protein synthesis. So actually, you already know, I already mentioned for that slide, G- gene expression means DNA will produce mRNA and then mRNA will produce protein. So DNA produce mRNA is through transcription process. So in this live, I will explain about the mechanism of the transcription. Okay. So firstly, what is mean by transcription? So transcription, I already say it is mRNA to DNA to mRNA, right? So inilah, this is the process of transcription. DNA to mRNA. So it is the synthesis of mRNA using information in the DNA. So it is using the information from the DNA, and DNA act as a template to produce mRNA. So mRNA actually is more shorter than DNA because not all information in DNA will be produced. It only choose some of the information in the DNA only. Okay. Okay, so in transcription process, there are three stages of the process. Firstly is about initiation. Next is about elongation. And finally, it is about termination. Okay, so what are these process mean? So initiation is the start. Start punya. I just give briefing. Initiation is starting process. Means that which DNA which part of the DNA that the prokaryote need to that the MR that the MR that the code of the DNA to produce a mRNA. So because if you know that DNA is very long, it is very long. The length of the DNA is very long, and I already say mRNA is shorter than DNA. So not all the information from DNA will be transcribed to mRNA. So only certain information from the DNA will be transcribed. So initiation process is the process to choose to choose which part of DNA which DNA information to be transcribed into mRNA. Dia nak kena pilih. Semua DNA punya maklumat kena akan transcribe kepada mRNA. Bukan. Hanya certain sahaja. So macam mana dia pilih? Inilah initiation process. Lepas initiation process, this is the this is the process where the cell itself will choose the DNA information. And then after he choose the information, it will proceed to elongation. Elongation means that it will synthesis mRNA. The mRNA nucleotide will be added will be akan bertambah. We're adding the mRNA nucleotide and mRNA nucleotide using the DNA as the template. Okay, after that, termination is the stop process because DNA is very long. Not all the information from the DNA will be transcribed. So it need a process. It need a stop area. Macam ni, kalau DNA, ini DNA, kalau maksudnya ini DNA, initiation is dia pilih mana satu tempat yang kena dia start untuk transcribe. Maksudnya dia start, example, is start at here. This is the start point. Yeah, he start at here. Okay, the transcription is start at here. Okay, transcription start. Lepas tu, next step is about elongation. So it will use this DNA template, begin to synthesize the mRNA strand. So I use different color. Okay. It will begin to synthesize mRNA strand. Okay, mRNA strand dah synthesize, but actually it needs stop. Dia kena henti. Bukan dia synthesize sampai hujung dunia. Bukan. Because DNA is very long. Dia bukan synthesize sampai hujung dunia. Dia akan stop dunia. Sorry. Dia kena henti punya. So macam mana henti? Through the termination process. So kadang-kadang lah dia stop at here. Maksudnya maybe it stop at here. This is the termination part. So the mRNA length of mRNA is like this only. This is the length of mRNA and only. So this is the whole process of the transcription. This is just the overview. So later, I will explain one by one about the initiation, elongation, and termination. And because I don't know, since if you according to the MPM syllabus, actually it only say that explain the syllabus just say that the candidate need to able to explain transcription and translation only, but it does not mention which cell you learn, because actually in the prokaryote and for the transcription in prokaryote and eukaryote are different. They are quite different. Not all the same. So if you refer to Campbell, mostly it will focus on eukaryote lah. But actually it got mentioned prokaryote, but no, that does not include in gambar lah. Tak ada gambar punya lah. Hanya dia just mention only, dia tak ada include gambar. So the gambar in the Campbell mostly is for the eukaryote one. But actually, if you see another genetic book, it will explain very detailed in the differences between eukaryote transcription and prokaryote transcription. So in my life, so I will explain some of the differences between transcription prokaryote and transcription for eukaryote. So I will proceed to prokaryote first, because prokaryote is more easier kan. Lebih senang untuk faham. Kerana eukaryote is more complex. Because eukaryote you need to know eukaryote got nucleus, prokaryote tak ada nucleus. So prokaryote is more easier compared to eukaryote. Okay, so for prokaryote initiation, so the main you need to know the main enzyme for the transcription is the RNA polymerase. The main enzyme to synthesis mRNA is RNA polymerase. You have to be careful. You cannot, if you write DNA polymerase, salah. DNA polymerase is for DNA replication. So for transcription, it is involved for RNA polymerase. Okay, so for prokaryote, firstly, they have to initiation process. They have to determine the position first, the initial position first. Which position of the DNA is need to begin the transcription? Siapa penentuan? Who determine the position? So actually in prokaryote we got one coding region. It is nucleotide region, coding region called promoter. So promoter will act as the I use different color. Promoter will act as the initial act as the direction or panduan macam panduan kepada RNA for the RNA polymerase to make sure the RNA polymerase will bind toward the promoter only. Dia tak akan bind to other parts one. It will bind to the promoter of the DNA. So what is the promoter? So the promoter sequence of the DNA for prokaryote got two types of sequence. Either it is negative 3,5 at the negative 3,5 region, which is known as TTGACG, and or negative 10 region TATAT. So these are the two nucleotide, two sequences that act as promoter one. Either one. Actually it is either one. Maybe it can appear in one DNA strand, maybe it can appear two. But actually either one or is okay already. Either one. Either it is TATAT or TTGACG. It is located normally is located downstream before transcription site. So you have to know the downstream and upstream process. Downstream terms and upstream terms. You have to know this. Wait apa lagi? Okay, you have to know the downstream and upstream. Okay, later I see. Later. Okay. Nanti wait a minute hold on. Saya kena tengok sikit my life. Cakap ada problem. My setting of the live. Okay, okay. Sorry. Tadi just now I set my live setting. Okay. So for now, you have to determine the upstream and downstream first. So because this one, I think Campbell tak ada cakap lah. Ni one memang Campbell dah tak ada cakap lah. So actually, if I let's say this is the start point, actually if locate to this is we call downstream. And if we it located at the left, it is known as upstream. So ini kalau kami panggil point lah, it is the starter point of the transcription. So if this is after point of transcription, you at the left hand side it is called as upstream, and for the right hand side it is known as downstream. So this one is at an upstream lah. So if upstream of the transcription starter point, so negative 10 region means that there are about 10 nucleotide ada 10 nucleotide dari sini 10 nucleotide 11 12 akan ada TAT. So this is the region of the promoter lah. So after the vagonite, after got promoter, so RNA polymerase will bind to the promoter. It will bind to the promoter of the DNA. So this is the places where the initiation process occur for prokaryote. Hanya for prokaryote only. So lebih senang sikit lah compared to eukaryote. Later I will explain about eukaryote one. Okay, ini untuk prokaryote. So senang saja. They have a promoter. So promoter will have these two sequence. Either it is TATAT or TTGACG that is located upstream from the transcription start site. After got this promoter, RNA polymerase will bind to the promoter. And then when it bind to the promoter, then it will proceed to the elongation process. Okay, that's this one I will explain all of the process for prokaryote first. So lebih senang sikit. So I will proceed to elongation. So elongation actually prokaryote and eukaryote all are the same. Tak ada perbezaan. Dah tak ada perbezaan yang banyak lah. So all are the same. So after RNA polymerase bind to the promoter site, then it will start the elongation process. So elongation process is the RNA nucleotide will be added lah. Will be directly added from the starter sites of the transcription, which is complementary to the DNA one. So only for transcription process, only one of the DNA will act as template only. So only one strand of DNA will act as template. Hanya salah satu sahaja, bukan dua. Hanya satu sahaja. Yang for like this, it is the for the codon region lah. For the non-coding region will be act as the template. Okay. Now so next I will proceed. So this codon region adding of the nucleotide will be complementary to the DNA template lah. Okay, so this one is a templation of DNA. This one will act as the non-coding. We as a non-coding region. So because coding region, the non-coding of the DNA will act as the template. Then it will form the coding of the mRNA. So this one, the RNA nucleotide will be added, directly added. So this is ada perbezaan dengan DNA replication lah. Because DNA replication we need primase first. It need RNA primer, but this one don't need lah. It can directly add. Dia tak macam DNA polymerase punya. RNA polymerase can directly add, directly produce the mRNA directly add into it. It will based on the complementary. So like this, if you see this, this one is T, right? So the complementary is L. So the DNA is A. So the complementary is U. So it will directly add the RNA nucleotide until it reach the termination process. And the direction of transcription is also 5 to 3. Right. 5 to 3 also. Same as DNA replication. Okay. Okay, so this is the elongation process, almost same as the DNA replication. It just that it is can directly add RNA nucleotide, and only one strand of DNA will act as template only. Hanya satu template sahaja act as the hanya satu strand sahaja act as a template. Okay, this is elongation. Next is about termination. So termination is what? Termination is a process of they have to stop adding the RNA nucleotide. So yang ni dia akan tambah sampai dia sentuh termination. So macam mana sel tu nak tahu RNA polymerase will know it already reached termination. Because the RNA polymerase need to release first. The RNA polymerase need to release, then the RNA nucleotide baru tak akan bertambah. If RNA not release, it will directly add until RNA polymerase is disappear. So how to make the RNA polymerase release from the DNA? So for the prokaryote, there are two types of termination. So the first type termination is called intrinsic termination. Okay, intrinsic termination is that when the RNA polymerase recognize there are the inverted repeat sequence in the DNA. Masuk macam ni lah. Inverted repeat, which is called what? If you see this, I use this. If you see that this one AGCT GAC. And if you see the strand at here, this one 3 to 5. So this one also 3 to 5, right? Kalau awak tengok sini. Lepas tu awak tengok dari sini. This one is 3 to 5 direction. This one, if you see from 3 to 5 direction, dari kanan ke kiri, you also see that AGC, TGAC, AGC, TGAC sama sahaja dengan ini. So this is what we call inverted repeat. It already repeat, but it is in inverted form. So actually kalau awak tengok direction, yang ni dari kiri ke kanan, yang ni dari kanan ke kiri, you see that it is actually the same. So if the RNA polymerase recognize this, and then after the inverted repeat, it already contain it will contain the sequence that is totally same. You see that this is all already T T T T T T. This one already A. So already all the poly you says that this is poly sequence lah. Poly A sequence. Kalau awak dapat tengok macam ni, this means that the RNA polymerase already reached the termination region. So at that time, the mRNA produced will become like that. Awak tengok akan menjadi macam ni. It will produce like that. So it will produce UCGA, UCUG, and then if you see that CAG, TGCA macam tak ada berkait because of the DNA. DNA already inverted repeat, so this one actually it is also inverted repeat. Inverted repeat. Okay, just that this one because if you use the yang bawah punya as template, awak akan copy ke sini, copy yang sini atas punya. Lepas tu because of this is as a template, akan copy yang bawah sana punya. Tengok. Okay, so because of the inverted repeat is occur, so the mRNA, the RNA produced, it will form hairpin structure because it already can already form hairpin structure already. Kenapa? Because of this. Because if you see that this one the A at here, you see A at here, it can be complementary to the U ke sini dari ini. Kan? A will U and G it will complementary to ke sini. Ini C ni akan complementary ke G ni. Sama sahaja ini, lepas tu yang ni, lepas tu yang ni, lepas tu yang ni. So it is complementary together already. Although it is RNA. It is already complementary in the single stranded already. So it is able to form the hairpin structure. Dia akan form hairpin structure lah. Followed by lepas tu after hairpin structure you can see followed by immediately new region downstream. So this one kalau mRNA because at that time RNA polymerase will move to the right hand side. Dia pun juga akan transcript akan banyak RNA nucleotide is added until here. The mRNA already form hairpin structure. So this one already will cause termination already. So at that time, RNA polymerase will directly release. It will directly release out from the DNA and then stop adding the RNA nucleotide to mRNA already. So this is the termination. So this will cause termination. Ini akan cause termination. This structure will cause the termination process. Kalau tak ada structure ni, RNA terus tambah lah. Dia akan tambah ke sini, tambah lagi. Because of this structure, it will directly prevent the RNA polymerase to adding the RNA nucleotide and release from the DNA, and finally your mRNA is produced. So this is the termination process. Okay, so this is first type of the termination for prokaryote. Ini prokaryote lah. Awak kena ingat ini prokaryote. Next is the second type of termination we call RHO dependent termination. Okay, RHO dependent termination, if you see this RHO, actually it is a type of the protein. It is a type of the protein component that can recognize the termination region, right? So RHO accompanies the RNA polymerase as it synthesizes the growing mRNA. So once the RNA polymerase had met the RHO recognization site, so RHO protein actually it will recognize it will follow RNA polymerase one, and it will once the RNA polymerase recognize the uh pass through the RHO recognition site. Maksudnya RHO akan bind dekat region tu lah. So RH will bind to this and cause a change in conformation of the RNA polymerase. Once the RHO protein bind to the RHO recognition site, it will cause RNA polymerase change the shape. The shape of the RNA polymerase will change, and RHO then untwist the newly formed mRNA, strand for the DNA, and subsequently the mRNA and RNA polymerase will fall off the DNA and RHO detach from the mRNA. So maksudnya kalau RNA polymerase pass through the RHO recognition site, the RHO protein will bind to the recognition site and it is activated and will change the shape of the RNA polymerase. So since the RNA polymerase shape is changed, those shapes will be altered. Will change since the RNA polymerase shape is changed, so it already cannot proceed to the hand side and already cannot to adding more RNA nucleotide. And then this RH protein will untwist the newly formed mRNA, means that it will try to separate the mRNA from the DNA. Dia akan cuba untuk potongkan mRNA tu, it will untwist at here like that. It will untwist the mRNA to make sure that mRNA can be separated from the DNA. Then finally, all of these component RNA polymerase, mRNA, and also RH protein will all of them will be released from the DNA. So this will become so the termination is N. So the adding of the RNA nucleotide from RNA polymerase is N. So normally, because and somebody will ask, so what is the RHO recognition site? So actually, there are no definite answer. But we only know RHO recognition site will contain more CG sequence one. It is rich in CG sequence. We only just know that because there are no specific meaning for RHO recognition site. Because so normally, but they found that just can simply meaning that RHO recognition site normally is rich in CG sequence lah. Kalau ada banyak CG punya, normally it is RHO recognition site, and then it will trigger the termination process and stop adding RNA nucleotide lah. Lepas tu mRNA will be released and newly mRNA will be formed. So this is the termination, another types of termination for prokaryote. Ini pun prokaryote juga. So yang ni is use RHO protein. Yang ni adalah bergantung kepada DNA strand. It does not use any protein one. Dia tak ada apa-apa protein law punya. It is according to the DNA strand. Kalau DNA strand have inverted repeat, and it will trigger termination. Kalau DNA strand tak ada inverted repeat and no termination occur. So sometimes if the DNA does not too long or does not have the inverted repeat, so normally the RHO will take over the termination process lah. So either two lah. The scientists found these two type of termination occur in the prokaryote lah. Okay. So this is all about prokaryote. Okay, ini prokaryote sahaja. Lepas tu, I will enter eukaryote. So eukaryote is animal cell and plant cell. Prokaryote is for bacteria cell one, right? Eukaryote is about the animal cell like us, and also the plant cell one. Or fungi also, fungi also eukaryote. Okay, for eukaryote initiation process, dia macam sama dengan prokaryote sahaja. Tapi, tapi, tapi yang sama sahaja. Okay, just that the promoter region of the eukaryote is we call as data box. Kami panggil data box. So it will have the sequence of TATAAAA punya. So why we call data box? Because it contains TATA. The sequence is TATA. DATA. So it is normally about 25 nucleotide upstream from the transcriptional start point. So this is the start point. Normally it's about 25 nucleotide upstream. So this one is upstream process, what I already explained. Okay. Okay, so this is the promoter of the eukaryote. Berbeza dengan ini lah. Ini adalah promoter untuk prokaryote. Prokaryote ini. This is for eukaryote. Eukaryote we call data box. Okay, start point lepas tu, and then there will be but before that, there was several transcription factor. Tak ada in eukaryote, no the RNA polymerase will not directly bind to the promoter one. It have the some transcription factor dahulu. First, compared to prokaryote. Prokaryote directly RNA polymerase will bind to the promoter one. But for the eukaryote, there are the some transcription factor will bind to the data box first. It will bind to the data box first. Before RNA polymerase 2 can be bind correctly. So this is to ensure that the RNA polymerase 2. So if you see this in eukaryote, you have to mention RNA polymerase 2. Dua punya. For prokaryote, only one. Prokaryote tak perlu mention. Because in prokaryote, only one type of RNA polymerase. But in eukaryote, there are 3 types of RNA polymerase. But only the RNA polymerase 2 were responsible to synthesis mRNA in transcription. Yeah, other types is another one. If I'm not mistaken, type 1 is for tRNA 1, but I already forget type 3. Type 1 is for tRNA. Untuk menghasilkan tRNA. But I don't know type 3 already. I already forget type 3. But for the mRNA is type 2. So you have to alert. For prokaryote, it is RNA polymerase 2. Awak tak boleh hanya cakap RNA polymerase saja. If you say RNA polymerase for eukaryote, salah. It is wrong. RNA polymerase only for prokaryote only. In eukaryote, you have to add 2 at behind. Ada 2 punya. Because eukaryote contain 3 types of RNA polymerase. Okay. Lepas tu, RNA polymerase 2. So, before RNA polymerase 2 can bind to the promoter region, some of the transcription factor will bind to the data box first. This is to ensure that RNA polymerase 2 can bind at the correct position. Because you have to know, for the eukaryote, it is located at the nucleus. And normally, the gene sequence in eukaryote is more complex to prokaryote one. Prokaryote is not complex like eukaryote. Eukaryote is very complex. So if no transcription factor, maybe RNA polymerase 2 will bind to the other part of the data box one. Because you need to know, in our nucleus, in our chromosome, our gene, does not contain only one data box. There are maybe several thousand, 10,000 type of data box. But not all data box will act as the transcription. So they have to recognize this data box first. To ensure that RNA polymerase 2 can bind correctly. So And then there were more and more additional transcription factor bagi signal lah. Macam signal lah untuk RNA polymerase tu. Kalau tak ada signal, maybe RNA polymerase tu akan bind, awak pun tak tahu dia bind ke mana lah. But this is act as the signal or bagi RNA polymerase tu boleh nampak, oh ini besar ni, ada signal yang besar. Bahawa inilah tempat yang saya kena pergi. Kalau tak ada signal, dia pilih nampak macam pening kepala lah, nak pergi mana ni, tak tahu. Tak tahu. So ada signal, baru tahu lah, macam kereta kan, awak pandu kereta, awak tahu, oh ini ada signal, saya pergi ke sini. Kalau tak ada signal punya, awak pun tak tahu awak nak pergi mana. So awak tahu destinasi awak. So awak destinasi nak pergi Ipoh. So you just ikut Ipoh saja. Kalau awak destinasi ke Ipoh, tapi awak tak ada signal, saya nak pergi mana ni? Kalau tak ada leader box, so awak pun tak tahu awak pergi mana. Maybe awak nak pergi Ipoh, awak pergi Alostar pula. Tak tahu kan, kerana tak ada signal. So there are the signal untuk for this also. So RNA polymerase 2 will know the signal so that it will bind to the correct sequence. So there are more and more transcription factor will bind to the DNA, then it will form the bind to the DNA and also RNA polymerase 2 will also bind to it. Banyak transcription factor as signal, lepas tu RNA polymerase 2 tahu, lepas tu dia akan bind into it juga. So di ini yang grey color one is the RNA polymerase 2, yang blue color one is transcription factor. So this one it will form transcription initiation complex. Dia akan form transcription initiation complex. Then the RNA polymerase 2 will break down the DNA double-stranded DNA double helix structure. This one I just forget mention at prokaryote lah. Actually prokaryote also need, because you need to know DNA is double helix. So then it need to unwind the DNA double helix structure first, baru boleh proceed to elongation. This is same as in prokaryote lah. So just now I forget to mention lah. I forget for mention for prokaryote. Actually it need to unwind RNA polymerase will unwind the double helix structure of DNA, and then it will baru boleh proceed to the elongation. So elongation step for both eukaryote actually it is same. Also, the RNA nucleotide will directly add the nucleotide complementary to the DNA and from the starter point lah. Dari sini lah. Dari sini. Lepas tu ini yang bawah punya add as template strand, and RNA nucleotide akan tambah dari sini lah. From 5 to 3 direction. Sama saja 5 to 3 direction. Okay. So elongation is same as prokaryote, but termination tak sama lah. Termination tak sama lah. So termination after adding the mRNA. So yang blue color is DNA, yang merah punya is RNA. This is mRNA. mRNA. So after the RNA polymerase 2, they're adding the nucleotide after the mRNA contain the vagonisation site called AAUAAA. AAUAAA. Ini kalau dia sudah ada sequence ini untuk mRNA, so there are the complex protein structure called polyadenylation complex. Polyadenylation complex akan bind to it. Formation lah. So ini kompleks ada banyak komponen buat. Dia ada banyak komponen buat. Dia ada protein, ada karbohidrat buat. Dia ada banyak komponen buat. Mainly is protein component lah. So this one polyadenylation complex, it will be formed and bind to this vagonisation site. So once it bind, this complex will cut down the mRNA. Dia akan cut down dari sini lah. Dari sini dia akan directly cut down. It will cut down this complex will cut down the mRNA. So this one is the mRNA strand. ini yang mRNA strain yang kami nak finally produce. But you need to know RNA polymerase still RNA polymerase still were adding the nucleotide. So this complex will change the shape of the RNA polymerase 2. And then another enzyme called azonuclease will bind to the RNA polymerase 2 and then cut down and release the RNA polymerase 2 from the DNA. So the RNA polymerase 2 finally it will release from the DNA and yang ni extra side of the mRNA will be hydrolyzed by this enzyme. Dia akan macam makan lah. Yang general term, it will makan mRNA ni jadi dia tak ada lah. Hanya ini sahaja this is the mRNA yang kami nak. So this is the termination of the eukaryote. So yang paling penting dia kena ada veganization kena ada sequence ni AAUAAA. Kalau ada ni dia akan form polyadenylation complex. Yang ni dia akan cut down the mRNA. It will cut down the mRNA. And then also will change the RNA polymerase 2 shape. So the RNA polymerase 2 cannot add more RNA nucleotide. Lepas tu other type of enzyme called azonuclease, it will release the RNA polymerase 2 from DNA and also hydrolyze the undesired mRNA. Okay, though this is the termination of eukaryote. So totally tak sama. If you see that it is totally tak sama punya for prokaryote dan eukaryote. So you have to alert lah. So I think that's all for the transcription process. Actually, transcription process is done lah. Sudah habis lah. So I hopefully you guys understand lah. So since okay, so I just make a repetition. So for initiation of prokaryote, so you have to know the transcription process, the main enzyme we use is RNA polymerase. For prokaryote, we use RNA polymerase only. But for eukaryote, we have to add 2 at behind. Ini for eukaryote. Okay, so for prokaryote initiation, they have to recognize promoter. So our promoter sequence is upstream from the transcription start site. So either one TAT, AAT, or TTG, ACG. So this is the promoter of the RNA polymerase. This is for prokaryote. So after it recognizes got this promoter, then RNA polymerase will bind to this promoter and unwind the double helix structure of DNA, and then it will proceed to elongation. This is for prokaryote. So lebih senang. But for the eukaryote, the RNA polymerase 2 is the main enzyme that we use in the transcription. And for the initiation process, the promoter region that need to be recognized is tata box, so TATA. Okay. Lepas tu, the eukaryote need to add the transcription factor to the tata box first. Dia kena ada transcription factor dahulu. Baru RNA polymerase 2 can add, because transcription factor will act as the signal to ensure that RNA polymerase can bind to the correct position and orientation. Lepas tu, RNA polymerase 2 tambah bind to the promoter region, and then it will start to synthesis the mRNA lah. So that's why they will form transcription initiation complex. And for the elongation process for prokaryote and eukaryote are same. It is in 5 to 3 direction. So it is same as the DNA replication lah. Kena ingat juga. Okay, so the RNA polymerase, either RNA polymerase or RNA polymerase 2 for prokaryote or eukaryote, it will directly add the RNA nucleotide to the RNA nucleotide which is complementary to the DNA. And for transcription, only one strand of DNA act as template. So normally, the bottom side we call as non-coding region. Non-coding region strand will be act as template one. So normally it must be complementary to the DNA and produce mRNA until it reach termination. And for the termination, there are two types of termination for prokaryote. First type one is when the DNA contain inverted repeat, where it where it, where it contain the inverted repeat and finally end with bridge of poly A tail for DNA. So the mRNA will produce the hairpin loop structure and cause the RNA polymerase to be released from DNA and the termination start lah, trigger termination lah. This hairpin structure will trigger the termination and RNA polymerase will be released from the DNA and termination and the transcription process is habis lah, stop already. So the mRNA already produced. And another type is based on the RHO dependent. So RHO dependent is that will recognize the RHO recognition site, which in CG sequence. After it recognizes, it will change the shape of the RNA polymerase and it will also untwist the mRNA, uh, ensure that the mRNA can be released from DNA also, and also the RNA polymerase will be released from the DNA, and finally mRNA is produced and transcription process is stopped. But for the eukaryote, it needs to recognize the sequence called AAU-AAA in mRNA, and after it recognizes the sequence, the one complex will form called polyannulation complex. It will form and it will cut the mRNA and also change the shape of the RNA polymerase 2, and then another enzyme called exonuclease will release the RNA polymerase 2 from the DNA and also hydrolyze the undesired mRNA strand. So this is the overall of the eukaryote lah, for the eukaryote process, and also overall of the transcription process lah. So transcription process, you have to know it is from DNA to produce mRNA. Okay, ini paling penting lah. The final product you will produce is mRNA. Okay, next. Before we go to the translation, but actually for eukaryote, it needs special for eukaryote only. Normally it's for eukaryote. Prokaryote does not have this problem normally. In eukaryote, the mRNA produced will contain exon in an intron 1. Kami ada exon dan intron 1. Apa itu exon? Apa itu intron? So, in general term, general meaning, exon is the coding part that we need. Intron yang kami tak nak punya. Kami tak nak intron because they do not code before the protein. The sequence for the intron it does not have the coding for protein. Hanya exon sahaja yang ada untuk code for protein. Because intron does not have any information that can be coded to produce protein 1. Because we have to know that our final product is to produce protein. Is to produce protein 1. So actually for eukaryote, since our DNA is very long, right? So even though mRNA is shorter than DNA, but our mRNA will be longer compared to the for prokaryote 1. For prokaryote. So in our mRNA, we also have exon, intron, and exon. Exon and intron region. And we want exon. Kami tak mau intron. Because intron does not contain any coding to any information that can code for protein 1. Hanya exon sahaja yang ada. Okay. Only exon yang ada. So sekarang kami ada mRNA. Tapi yang ni we call as pre-mRNA lah. Yang ni pre sahaja. Kerana tak ada, yang ni tak ada, yang ni ada intron. So we have to remove this intron. We have to remove this intron first. Baru kami akan hasil mRNA yang baru. Okay, so if you see this picture, you see this pre-mRNA, dia ada kuning-kuning sini kan? Yang ni, kuning punya. Lepas tu yang ada ni. So, So, yang ni kuning is the requirement to remove intron. So, if you want to remove intron, we will known as RNA processing, called RNA splicing. So, this is the scientific terms for the RNA processing to remove intron. So, if we want to remove intron, we have to add two things first. First one is the cap. Second one is poly-A dia. Poly-A dia means semua yang ni A punya. Semua A punya. Yang ni adalah cap, satu cap. Okay, so we have to adding the cap first and also the poly-A dia baru boleh buat RNA splicing. Kalau tak ada dua ni, the RNA splicing is cannot be happen. So we have to add these two things first. So addition of the gap. So what are the gap? So the gap actually is known as guanosine triphosphate, GTP. GTP. Inilah gap dia. So this is the RNA nucleotide, nucleic acid one. Basic unit of nucleic acid. Kan? Ini dua ni. Yellow punya dan juga kuning color one. This is the 5' region. 5' one. So normally our gap is adding at the 5' region one. 5' region means that there are the 5, yang ni kan, yang ni 1, 2, 3, 4, 5. At the 5' end region, there are the gap. It will link with the gap. So the gap is known as GTP. So GTP will be added to the free end of the 5' region of mRNA. Kenapa dia call GTP? Because there are 3 phosphate group and that means this is the guanosine structure. Ini adalah guanosine structure. Guanosine structure, and then you will see that there were methyl group at here. Ini ada satu methyl group at here. So this is very important. Ini very important. Okay. So this is addition of gap. So our gap is GTP lah. Kena tambah satu GTP dekat 5' end of the mRNA. So gap ini. Lepas tu is addition of poly-A tail. So how to add our tail? A tail. So actually this is involved in termination process one. Kalau awak tengok, dia macam termination process. So this is the yang ni lah. Faham kan? Polyadenylation complex. After mRNA produce, it recognize AAA. Polyadenylation complex is occur. Kan? Ini termination process. Kan? Kan? So this polyadenylation complex, I already said it will cut down the mRNA. Dia akan cut down the mRNA. One. Lepas tu dia cut down. In the polyadenylation complex, actually 15 adenylation this complex got poly-A polymerase. Inilah enzyme untuk responsibility to add the A nucleotide into it. So after this complex cut down the mRNA and also release out the RNA polymerase 2, then the poly-A polymerase in this complex will begin at series number of poly-A tail lah. So it will form poly-A tail. Inilah. So it actually it will involve polyadenylation complex. So why? Because the poly-A polymerase is within this complex. So after it cut down the mRNA, so that it will form poly-A polymerase at here. Ini semua AAA lah awak. You can imagine lah yang ni lah semua A semua A punya. Macam ini lah semua A punya. So macam ni lah awak akan dapat semua A punya. So this is the process to addition of poly-A tail. So this is dia ada abang-adik untuk berkait dengan termination process lah in the eukaryote one. Okay. So after we adding these two component into mRNA, barulah kita boleh buat splicing process. So intron will be cut out and exon splice together. So splice means that akan gabung. Kan awak sudah cut kan? Ini ada lubang kan? Takkan. So kena gabung together. it need to gabung together. So akan jadi macam ni, hanya satu akson sahaja. Intro semua sudah hilang. Okay, macam mana? Macam mana? RNA splicing macam mana? So there were one complex called spliceosome. So called spliceosome. Dan yang ni lah, it is the complex that can play the major role in RNA splicing. So it is the one complex and have many ada banyak benda punya. So it has the contain protein called ribonucleoprotein. We label it U1 to U6. Inilah SNURPS. Kami in scientific short form we known as SNURPS. So it is yang ni semua spatial cell. So SNURPS there are U1 until U6. So U1, U2 tapi kami tak ada U3. We have U2AF saja. Dia tak ada U3 lah, tak tahu. Mereka label hanya U2AF. Lepas tu U4, U5 and U6. So these are the complex will be able responsibility in RNA splicing. Okay, so macam mana? So ini proses dia lah. Susah kan? Nampak macam susah. Tak boleh. Awak in the syllabus. It is in the syllabus. But this one is not take from Campbell book. This one is take from another genetic book. Actually, Campbell pun ada mention RNA splicing juga. Tapi saya rasa Campbell dia tak explain terlalu detail lah. Dia tak explain detail. Kalau awak baca Campbell, maybe awak akan ada kekeliruan lah. Tak tahu dia cakap apa. So maybe you can read other genetic book or you watch my video. You watch my video, then you understand. Okay, so U1, I already say spliceosome got U1 to U6. So first, if you see this, dia ada axon, intro, axon. So intro ni dekat tengah-tengah. So firstly, U1. So this one actually I label ini 5 point, ini 3 point. Normally 5 point here, 3 point here. Normally. So U1 it will bind to the 5 point side of the splice site. So actually we call a spike got the splice sites between the splice site. Kami ada splice site between the axon and intron. We got the splice site. Yang ni hijau-hijau punya. And then yang ni tengah-tengah punya ini kami panggil branch site. Ada meaning mereka, ada kerja mereka punya, bukan hanya label shock saja, ada kerja mereka punya. So this U1 and U2AF, this one is called U3 lah. Actually it's U3, but they don't use U3, they use U2AF. Okay, U1 and U2AF will bind to the splice site. So U1 is bind to the 5 point N1. U2AF is bind to the 3 point N1. After they bind U2 another spatial ribonucleoprotein U2 will bind to the branch site. So branch site at here, yang tengah-tengah punya. Awak tengok yang dia tekan yang ni punya. So akan bind dekat sini. Okay, after mereka bind, U4 and U6 akan bind dengan U2. Dia akan bind dekat U2. U2. And U5 will bind to the downward from the axon. Downstream of the axon. Downstream, what is mean by downstream? Because axon at here, dia akan bind dekat yang belah, yang kanan punya lah, kanan punya very kanan punya. Actually it's very right one. Very right one. Right part. It is very right part. Sangat kanan punya. Lepas tu after they bind, you will see that the splicing already occur. Lepas tu awak tengok yang ni U1 and U2 it will begin to come to each other, and U5 will push the strand from right hand side to left hand side. It will move. U5 will move from right hand side to left hand side, and then it will form the loop structure macam ni. Awak tengok dia akan form the loop structure macam ni. Macam ni. Macam ni. Macam ni. This is formed from U1 and U2 and also U5. And then, after that, U1 and U4 will be released lah. Mereka tak ada function. U1 and U4 will be released. Will be released. Will be released. Will be released. Released. And another, so U1 sudah release. So, U6 akan gantikan tempat ini. U6 akan gantikan tempat ini. Dia akan gantikan tempat ini. So, inilah role of the ribonucleoprotein habis. Tak ada cut lagi. Nanti ada final step. So, inilah final step. Inilah final step. So, after they form the loops like this, I will see that like this, the intron part, the intron part yang ni, we call splice site of the 3' one. 3' or splice, not 3' 5' sorry. 5' or splice site, it will bind to the branch site. Dia akan macam ni lah. Awak tengok dia akan macam ni lah. Kerana dia sudah ada loop structure. Lepas tu loop structure, it will bind to the branch site. Tengok macam ni lah. Macam ni lah. So you have to know U2, actually U2 is the part of the branch site. Lepas tu U6 ni, dia akan bind toward the U2 one. Dia akan bind lah. Dia akan cut down the binding, cut down the way of the, macam jalan lah kan, dia ada jalan kan, dia sudah macam ni. Okay, dia sudah macam ni. Ini adalah splicing site of 5'N. Dia akan cut down lah. Macam ni. Ya, macam ni. Dia sudah akan cut down lah. Cut down this, and then dia akan gerak macam ni. Bind ke kat sini. So, dia look like macam ni lah. Band side. Lepas tu, the 3'N slide, dia also akan cut down also. So, for A1, so 2 exon interviewing, so the 5'5 is cut first, and the 3' of the intron will look back to bind the band side, and then the 3' side is server next, and next end of the 2 exon are joined, the intron is released as a labiat structure. So we call as labiat structure. So ini pun dia akan cut lah. Lepas tu dia cut. Yang ni semua structure ni dia akan release. Lepas tu exon dan exon ni akan gabung together. So to form ini. So the main idea of this spatiosome is to form the loop structure first. Dia nak form loop structure. Form the loop structure so that it can cut and release in the form of labiat structure. The main function of this ribonucleoprotein. Dia form loop structure, lepas tu dia akan cut in form of labiat structure. Yang ni saya suka. Yang ni semua after dia cut, firstly it cut sini. Lepas tu form this, and then it cut ke sini. So semua ni structure, yang ni semua akan release ke macam ni. It will release at this side. So this one is called labiat structure. We know as labiat structure. And then after that, exon 1 and exon 2 will splice together. Exon 2 will splice together for ni lah. So inilah mRNA yang kami nak. mRNA yang kami nak. So for this loop structure macam mana form? Ini. Yang ni lah. Yang ni spliceosome yang form punya. So kalau tak ada spliceosome, yang ni tak akan berlaku. Kalau ini tak ada berlaku, memang tak ada splicing occur. So inilah sangat important. So until here it already stopped. Splice zone is already stopped. Later it will cut down the splice side, for 5' end, and then the splice side will be bind toward the bond side, and then another 3' splice side is cut. Later it will be released in the form of larvae structure, and then the exon 1 and exon 2 will be spliced together to form the newly formed mRNA. So this is the mRNA yang kami nak lah. Bukan yang ni lah. Ni kami tak nak. Pre-mRNA kami tak nak because got intro kami tak nak. Kami nak yang exon sahaja. So inilah konsep RNA splicing. So hopefully you guys can understand lah. Kalau kamu tak understand, maybe you can post YouTube lah. I hope you guys all can understand lah. So you just need to remember the main objective of the RNA splicing is to remove the intron. Kami tak mau intron. Kami tak mau intron. Kami nak exon sahaja. So this is the main role of the RNA splicing lah. So hopefully you guys can understand lah. What is for today's topic, we already learned transcription and also RNA processing. So for the last live for today, 9 o'clock, I will 8:30, not 9 o'clock, 8:30, I will explain about the translation process. So okay, the live I will hold on, is end here. So hopefully you guys can scan this feedback form so I can know my performance value lah for this live. So thanks for watching and your appreciation. So I apologize to that you can listen my live lah. So hopefully you can get the beneficial from this live. So thanks all and have a good day.